Okay, hello everyone, and thank you for joining us to this ISCB-SC webinars. Today, we are initiating the seminar sessions for our virtual seminar series. This is a program from January till April, and I would like to remind you, if you want to know more about the upcoming webinars or how you can present your research and be part of this initiative, you can follow us on Twitter as @ISCB-SC. So today we have a great talk to start with this seminar series. We have Alberto Santos. Alberto has a background in computer science and a master in bioinformatics. So he did his PhD in Denmark, where he developed several biomedical databases and used network biology to study diseases, to have a more holistic view of different biological patterns or diseases that he is studying currently. Afterwards, he continued at the Novo Nordisk Foundation Center for Protein Research, also in Copenhagen, as a postdoc in the clinical proteomics department, where he continued working with networks applied to proteomics and metabolomics data. Mainly, he is involved in the data analysis, that the data that comes from different kind of proteomics technologies. And Alberto is currently a group leader at the Center for Health Data Science at the University of Copenhagen also, and at the Big Data Institute in Oxford University. And he will present today his group, the Multiomics Analytics Group, and how they use graphs, to investigate diseases and identify diagnostic and prognostic markers. So Alberto, thank you very much for being here today with us. The floor is yours. Thanks.
Thank you very much. I'm really happy to be presented today. Let me see if I can just start the presentation, and please let me know if it works. I take it as it works, right? Okay, so thank you very much for the introduction. You see,
sorry, Alberto. I think that we have a problem with the monitor, maybe, because now we are seeing in the presenter mode. We can see the two slides. The slide
I will stop sharing and then share again. So if I do it like this, it should work.
Yeah, now it's okay. Thank you.
Perfect. So again, thank you very much for the invitation, and thank you very much for the introduction. It's always an honor to present, but today it's even more. I was invited by Yesid, and he's quite an important person in my research career, so it's always good to have him on board. And today I would like to present the Multiomics Analytics Group and how we are using graphs to try to define diseases and by integrating multiple omics data types. So first of all, a small introduction. I'm from Spain. I was born in Valencia, where I did my studies in computer science. And then after quite a bit traveling around and working also in industry to try to fix all these computational skills, I decided to move into academia, and I took a master's in bioinformatics in Barcelona, where I had the opportunity in the second year to do my master's thesis in Copenhagen in the lab of Lars Juul Jensen. And at the end of my master's thesis, I was offered a PhD position. So I took it, and I worked with networks, and I developed different databases. And then I got to love how networks help you simplify the complexity of biology and the complexity of disease. And then after my PhD, I was also offered a postdoc position in the same center at the Novo Nordisk Foundation Center for Protein Research in a different department, in the clinical proteomics department, that was mainly a wet lab. But I decided to join because it was a great opportunity to get the best data that was generated in clinical proteomics, and it was really like that. So I enjoyed very much working in this lab, and I worked in trying to interpret the proteomics data in order to bring it into the clinic. And at the end of 2019, I was offered the possibility to open a lab in the Big Data Institute in Oxford in the UK. And it was one of those opportunities you cannot say no. And it was also in close collaboration with the Proteomics Facility and the Target Discovery Institute. So it was a great opportunity, and also opening your own lab, it was really amazing. But then I started in April, but then as you all know, April was a kind of difficult month. So even though I started in Oxford, I stayed in Copenhagen and I worked remotely. And then in July, I had the opportunity to open the lab also here in Copenhagen. So now the Multiomics Analytics Group is located in both places. It's at the Center for Health Data Science at the University of Copenhagen, and also at the VDI in Oxford. And what you can see on the screen is more or less how the group will look in the next few years. And we are now currently hiring 2 postdocs that will be one in Copenhagen, one in Oxford. And the idea is that in both locations we will have our different projects, but also common projects, and we will use the locations and collaborations with different hospitals to try to validate and also confirm some of the findings that we get with our methodologies. The Multiomics Analytics Group is growing around the Clinical Knowledge Graph, which is a framework that I developed, or that we developed during my postdoc years, and we are extending this framework to integrate more data types and also to include machine learning in the process. And we want to focus on infectious diseases, biomarker and target discovery, and also treatment recommendation. When I talk about data integration, I'm talking about all the different omics that you have out there, so proteomics, metabolomics, lipidomics, and so on. And then also other types of data like imaging, sequencing, and also electronic health records. And the concept of graph machine learning, we will use it to summarize knowledge, predict new links in the networks, and also build similarity networks. And ultimately, the objective is to translate those results into something that is actionable in the clinic. So it is a translational bioinformatics lab, and when you want to translate omics data or multi-omics data into the clinic, and you need the results to be clear, interpretable, actionable, and quick. And I always give the example of biochemical blood tests, where you have different analytes that are measured, you know the ranges of normality, and then anything that is below or above triggers and diagnosis, and then that can lead to an intervention, which could be a diet, exercise, or drug treatment, or surgery. But when you are working with multidimensional data, heterogeneous data, then the bottleneck is in the interpretation. If you want to find those results that are actionable in the clinic. So if we think about the data like this, like a box with different shapes and different types, what we are trying to do is trying to connect those different data points to find how they are connected, so why we find them relevant in our experiments. And those connections could be protein-protein interactions, pathways, functional associations, and many other different links or types of links. Those are the connections, and when you look at this, you can see that it's already a graph where you have different components or nodes and different links among them, or edges. So those are graphs, and the beauty of graphs is that they allow you to define a system of interconnected components that provides a framework to immediately integrate heterogeneous data. And then you have all the graph theory behind it, so you can reveal the topological properties of some of the components, like the role of a specific nodes, how the information flows, on how robust the system is when you remove a node or you remove one of the links. And this is what is known as connective knowledge. So you extract knowledge from the connections. And to give an example, if we have a proteomics experiments, you will end up with a list of hits or proteins that are differentially regulated, for instance, in your system. And then you know that protein A is regulated. And then if you add on top knowledge about where it is expressed, you can see that it is expressed in the liver and present in blood. Associated to diseases like alcoholic fatty liver disease, liver cirrhosis, type 2 diabetes, and annotated in pathways, the fibrosis pathway or inflammation process. If you have, and on top of that, you have evidences of these associations in different publications. So if on top of that, you have another protein that is regulated, and you want to understand why and how they are related, then you can include protein-protein interactions. You can again see what are the annotated pathways and where this protein is expressed, and based on this, define that probably that this protein might as well be associated to the same diseases. So you can draw conclusions from this graph immediately. And this is a very dummy and naive example where you have only few nodes, and we could infer those links just based on the few links that we have. But generally, what you have is something like this, and even worse, where you have multiple nodes and edges. But still, graph theory allows you to perform some analysis immediately on the network. That could be clustering, for instance, that define communities of nodes that are interacting or linked more than with the rest of the network, or centrality, which reveals the relevance of some nodes to, for instance, to how the information flows and so on. But where is all that knowledge? So there is an exponential growth in the data generated without high throughput technology, and it's in genomics, like transcriptomics, epigenomics, and so on. But we are also collecting multiple layers of information, medical records, images, physiology, and also exposure and environmental data. And science is a cumulative process, so we share everything that we generate so that others can build on top. And we generally do that through publications. So there is plenty of information, relevant information in publications, and also data standards are allowed sharing and making the data accessible through biomedical databases. And as you can see in graph on the right, the number of publications of new biomedical databases is growing quite a lot. And this is only in one single journal. So why not put in everything together, the concept of graphs and also all the knowledge out there, into one single framework? And this is what we did in the clinical knowledge graph, which is a framework to automate the analysis of proteomics and metabolomics data and to try to accelerate interpretation as well. In order to automate the analysis, we needed to standardize all the data, and for that we needed to adapt to the different processing tools out there. And also we need to define ontologies or use ontologies to define the components and nodes in this graph. And then once we had this graph, we could use this connective knowledge to collect it from biomedical databases and also extract it from text mining of scientific articles to try to help in the interpretation of the data. So generally in a proteomics analysis workflow and in many omics as well, you have matrix with M proteins in this case and samples. You run sequence of statistical analysis, and you define a list of proteins that are relevant in your study. And then you try to annotate all those proteins using different databases to associate them to drugs, diseases, biological processes, pathways, publications, and so on. In the clinical knowledge graph, what we do is we have the same matrix that goes into the clinical knowledge graph. It is automatically analyzed, so it generates the list of hits immediately, and then tries to annotate with all the same types of the data that you saw before, drugs, diseases, and so on, the hits that we identified in our study. And once you have all this data together, it tries to summarize it nicely so that it hopefully can help in the interpretation. And we can do this with proteins, and we can do this with metabolomics data as well. It is this different matrix, and instead of proteins, you have metabolites, and then just goes in, you run different statistical analysis, and then you annotate with all the knowledge that you have in the graph. So the clinical knowledge graph builds a database directly in the shape of a graph. And in this database, we have experimental data and also biomedical data from different databases, and all annotated with ontologies. And then we automate the analysis workflow because we understand how the data goes into this graph structure. And that way, we hope that we help finding information in the graph, and also we add on top these relationships that can allow us to find hidden patterns, such as clusters, correlations, causality, and so on. The clinical knowledge graph is built with two main components. One is the database, the graph, the knowledge graph, that gathers all this information. And then it's all the Python code that we build around so that you can generate a statistical analysis and visualizations of the results. When you build a knowledge graph, one of the most important steps is the definition of a data model, which defines which nodes and relationships go into the graph. And in this case, you can see our data model, and you can see that we have in the model, we have proteins, we have genes, transcripts, somatic mutations, peptides, clinical variables, metabolites, and so on. And this defines all the data that goes in, and it allows us to integrate experiments and biomedical databases. We integrated 26 databases, and we use 9 ontologies to define this kind of the vocabulary that we have in this knowledge graph. Currently, we have more than 19 million nodes in the graph and more than 200 million relationships that we can query using the Cypher query language, which represents more or less with text the structure of a node, two related nodes. Sorry. So the CKG workflow changed a little bit the normal proteomics or omics workflow. We expect that the scientists will start by creating a project directly in the clinical knowledge graph in the app, and that will generate an identifier that can be used across your experiments. And once the data is ready, you just upload the experimental design, which defines what are your samples, or in this case, what are the subjects, what are the biological samples you got from the subjects or patients, and then the analytical samples, what you used in the mass spec sequencer, and so on. The clinical metadata that you collected from the subjects, and then the proteomics data sets in one of those formats, tabular format, or one of the software tools used to process the raw data. That goes into the graph, and automatically you generate an analysis with quality control, differential regulation, and so on. And that's run automatically based on a definition of a configuration, or based on a configuration file. But if you want to do something special, what we've done is we coupled with Jupyter Notebooks, which allows you to use all the libraries that we generated to run more analysis and also extract specific information from the graph. This is the configuration file, or how it looks. It's basically a YAML file where you define the different sections in your analysis, and for each of the sections you define a type of analysis and a type of visualization and which attributes or parameters you use. And then in some of these analyses, you will generate new data sets. Like in the case of principal component analysis, you will generate a table or a data frame with the loadings for each of the proteins. And in the ANOVA, you will generate a regulation table where you have the different comparisons and what the values for the fold chains and p-value and adjusted p-value are. And if you were to add a new function into the clinical knowledge graph, you could just add a new line in the configuration file, and that would simply add a new analysis into the automated pipeline. So this is the way we are working, and these are all the different functions and visualizations that we have currently in the clinical knowledge graph, and this will continue growing with new developments. We've been using the clinical knowledge graph in different studies and defining whether we get, so these are published studies, and we are trying to find out if by using the clinical knowledge graph, not we can replicate the results that were obtained, but also identify new ones. So I will introduce the first one. It's a non-alcoholic fatty liver disease. It's a biomarker discovery study using plasma proteomics. And basically, you have your matrix with proteomics data, and so the quantification of the proteins for the different samples, and we have in this case different groups or disease groups: cirrhotic, non-alcoholic fatty liver disease, non-alcoholic fatty liver disease, and type 2 diabetes, and type 2 diabetes. And then it will automatically run a sequence of analysis that we have defined, where you get quality control plots, differential regulation analysis, correlation analysis, weighted gene co-expression network analysis that allows us to correlate the clinical variables and the proteomics or the protein profiles. And then we annotate with the different components we have in the graph: diseases, drugs, complexes, metabolites, publications, and so on. And we were able to reproduce the results that were already published, but also identify relevant things like protein complexes that were not identified in the first analysis. The rectal carcinoma is a treatment recommendation study where we run proteomics and exome sequencing, and the idea or the objective here was to try to find a treatment that was that could be used in the patient, and for that we ran proteomics, and then we defined a pipeline that we can reuse later on in other studies, where we take the upregulated proteins, we find drugs that inhibit those upregulated proteins, we profile those drugs and identify which of those drugs have less side effects, and then we looked for commencing of several of those drugs together in publications, so that we could recommend a combination of treatments. And then we came up with a combination of treatments that could be used, that could have been proposed in this case. This was defined in a Jupyter notebook, and what we are planning to do is every time we have a case like this, we can rerun this Jupyter notebook just by calling a different project or getting the proteomics data from this new project and then running the same pipeline to try to identify this or to propose a treatment. And there can be many improvements to this pipeline. So once you define a pipeline that works, that provides good information, we could add it to the configuration file, to the automated analysis in cases where what we are interested in is getting a treatment out. This, as I mentioned before, in this case study, we worked in a Jupyter notebook, and we coupled CKG with Jupyter notebooks and defined two different directories that can be used to understand how, on the one hand, how CKG works, looking at different recipes, like in this case, how to download directly data from Pride and analyze it afterwards, or reports where we reanalyze published studies using CKG. Like in this case, we looked at white and brown fat and what were the protein differences, and we defined also if there were any drug targets among the differentially regulated proteins. The number of recipes will continue growing, so we will keep showing functionality in the clinical knowledge graph, and we will continue adding more reanalysis of published studies so that it can give examples of how you can reanalyze your own data. One of the recipes we have currently is the knowledge summarization that we are using in the knowledge graph. And basically, you can provide a list of proteins. It generates all the knowledge based on the data model, so all the associations we have for this list of proteins, and then it uses centrality, betweenness, closeness, and so on, or PageRank to define or prioritize the nodes in the network. And it comes up with a subgraph of the whole knowledge graph that contains the most relevant information for your list of proteins. And it tries to identify which disease is the most relevant one, or what are the drugs that target more than one single protein, if there are any biological processes that are common among the list of proteins, and so on. And that's the graph that you can use to afterwards interpret what is happening or what is going on with the list of the proteins you provided. It allows you to specify a disease so that you can limit a little bit the graph to only associations to that disease and drugs that are used indicated for that disease. But in general, you can use it without the list of diseases so that it should already identify which disease or which list of diseases is related to your list of proteins. Then if I go back to the title of the talk, the defining disease using graphs, well, first of all, you can define diseases in many different ways, and here I'm providing 3 different types of definitions. One would be just the simple definition, text definition, and here I'm defining, or I took this definition from Mayo Clinic, which defines Alzheimer's disease as a progressive neurologic disorder that causes the brain to shrink, atrophy, and brain cells to die. But then you can also define it as in a molecular way by mentioning that there are 5,349 proteins associated with Alzheimer's disease, according to Open Targets platform, or there are 1,729 pathways related to Alzheimer's disease, according to Reactome database. But why not putting everything together into one single graph, where you define a disease based on the symptoms this disease has, the drugs that are indicated for this disease, the proteins that are associated with the disease, and metabolites, and in what tissue the disease happens, and what pathways are the proteins and metabolites annotated to. If you are able to build a graph like this for every single disease, then you can transform this graph into a vector using graph or node embedding. And then you can use these vectors to just build, for instance, a disease similarity network, where you just compare the different vectors using cosine similarity, for instance, and then you define what diseases are similar to what other diseases, and then you could look into which of those are comorbidities and which of those are confounded by indication, and so on. But the idea is that you use all the information you have for one single disease to build this vector, and then that's what you compare, so that you are not simply comparing the proteins they are associated to or pathways, but comparing everything you know from all diseases. So this is the part that we are trying to extend now with the graph machine learning, and it's basically learning representations from graph structures, and for that you can use the graph topology as features, you can use embeddings, or you can use graph neural networks, and this is what will come up or what we are working on currently in the lab. And we plan to use it for diseases and drugs, but also hopefully later on single patients, so that you can build a specific graph for each patient and then compare these graphs in order to identify which patients are similar to which patients, and hopefully from that recommend treatment. So if we go back to how we were representing data and trying to input knowledge in it, and then here we are trying to identify insights, and ultimately what we want to interpret this data to bring it to the clinic. The clinical knowledge graph is available in GitHub in this URL, and basically what you need is a Neo4j database, or it will generate a Neo4j database with all the data that we have in the data model. It will include all the recipes and reports in the Jupyter notebooks, and also you will be able to run the app that generates the automated reports. This is more or less the structure of the clinical knowledge graph, or the structure of the code. And here you have the graph connector and graph builder that allows you to reproduce the knowledge graph, the report manager that will generate the statistical reports, the notebooks with the recipes and reports, and the analytics core, which is the core of the clinical knowledge graph, with all the functionality to run the statistical analysis and also visualize the results. There is also a Docker container that will open 4 different ports, one with the Neo4j database, and you have 2 possible ways of accessing, either accessing through the app or the interface, or programmatically using the second port. Then you have the Jupyter Hub to open the Jupyter notebooks, and you have the CKG app in the port 8050. So basically you build the container, run it, and you get everything in one go. And with that, I'm finishing my presentation, and I would like to thank the Novo Nordisk Foundation Center for Protein Research, the Clinical Proteomics Lab. Most of the work was done during my postdoc time in this lab. And I would like to thank Matthias Mann, Narita Colasso, and Anna Laura Bach-Nielsen, who worked intensively also in the knowledge graph. And then I would like to thank Philip Charles, a senior postdoc in Oxford and the face of the group in Oxford at the Big Data Institute. And with that, I would like to thank you again, and I'll be happy to take any questions and hear some details of how to contact me if you're interested in working in the lab, visiting, or just have any issues with the knowledge graph or any requests. So I'll be happy to answer questions.
So thank you, Alberto. It was a really nice presentation. So thanks a lot. So we have time for questions from attendees. So please feel free to write in the chat box available on YouTube. So questions?
I'll keep the screen just in case.
Okay. So, but in the short time, I would like to ask you about the final users of your clinical knowledge graph. I mean, you mentioned that the reports are available on Jupyter notebooks. How stable are that Jupyter notebooks to display a lot of data? And also, if this clinical knowledge graph is going to be available to physicians, maybe in the future, maybe what do you think? Or maybe could you please describe us the challenge to share this info with physicians, and what kind of resource are you thinking to create in the future to make available this info for the physicians?
Yeah, so There are a couple of questions in your question. So the first one is, who are the main users right now? So we are thinking about two different types of users. On the one hand, I would say biologists, but anyone interested in just looking at quickly at how the generated data looks. And these users will access the automated reports, and we might play around with some of the parameters to change, for instance, how the principal component analysis is displayed, or what are the thresholds in the ANOVA analysis, and so on. But then we are also thinking about data science users that want to extend a little bit the analysis and go a bit beyond what is the automated analysis. And for these type of users, we recommend the Jupyter notebooks, where they will find, on the one hand, all the examples that they can reproduce with or replicate with their own data, but they can also extend using all the Python ecosystem, R ecosystem, but using the data generated from the graph. In the future, what we would hope is that we can generate a framework that it's able to digest so much the data that it can augment the decision making in the hospital, and for that it has to simplify the results quite a bit. So the doctors are, first of all, they won't have time to go through all the options and so on. They will need something that gives an actionable result. And for that, I think that's in the long future, because we still need to summarize the knowledge that we extract. We still need to understand what comes out and how to interpret it so that it can be actionable results. But in some cases, like for instance, the drug recommendations, treatment recommendations, it might be possible once we have enough data in the graph.
Cool. Thank you, Alberto. So we have now questions from Isse. So let me read. Okay, do you think that it's possible that there can be a kind of unified biomedical knowledge graph so far? So far, I understood that people individually develop them, which can be rather redundant effort.
Yes, so it is definitely a redundant effort, and I'm sure that it could be possible, it could be doable to have one single knowledge graph that everyone uses, and everyone, maybe not one, but many different ones, but at least that similar communities are using the same knowledge graph. And the idea with having the clinical knowledge graph open source goes in this direction, that we put it out there not only for people to just use it, but also to keep developing, implementing, and keep growing this, so that it can become a community effort instead of one or two labs effort. But it is difficult, like anything else in science. We try to work individually instead of in a community, with community approaches. And I'm sure that there are many things in the clinical knowledge graph that are not built as a community effort or with the community effort in mind that could be improved. But that's why we put it out there, so that it can there might be someone out there that can improve this part or change it completely.
Okay, thank you, Alberto. So we have.
And if I can just add a quick thing, so we will benefit if we have one single knowledge graph, because then we will have enough data to do all the machine learning that we want to do afterwards.
Okay, we have another question from Keren from Turkey. I wonder, do you integrate genetics data into clinical knowledge graph? I mean, genomics, transcriptomics, or epigenomics? Is CKG weighty based on proteomics?
So for now, because I was working in a proteomics lab, the focus of the clinical knowledge graph is on proteomics data. So all the parsers for different processing tools is focusing on proteomics and metabolomics data, but then the idea or the data model was built so that genomics data can be easily integrated as well. So if you have a standard format for transcriptomics, then and you build a parser for this transcriptomics data set that generates a file, a TSV file with your transcript and your quantification, that can easily go into the graph, and then all the statistical analysis can use this data set.
Okay, cool. So we have the second question from Ise. By putting different types of data into graph, how should one deal with continuous data? Example, expression levels, for example.
So the intensities or expression levels are attributes of the edge. So we have the analytical sample is a node, and then the analytical sample you will have many links to different proteins, transcripts, or genes, and in the edge you will have an attribute that is the value or the quantification. And in cases where you have multiple time points, so longitudinal data, what we are doing is we are including those will be different analytical samples, but the analytical sample will have another link to a time point that specifies when this analytical sample was taken.
Okay, so now Alberto from Chile. Hi, what about generation inference of new edges? Can your tool do that efficiently if there's nothing available, papers, PPIs, or is this what you are trying to solve with machine learning?
So that's part of what we want to do, and what we are doing is based on similar graph structures for a specific node. We can generate a link. Well, we can predict links based on these graph structures. And you can do this in different ways, but the database that we are using, that is Neo4j, provides a library, a graph data science library that allows this kind of predictions, link predictions. So you can do it directly in the database, or you can extract the data and then do it in Python, in our case.
Okay. So Daniel say, I'm very impressed. You mentioned the graph embedding to find similarities between different diseases. Do you think this can also be done for disease subtypes?
I don't know exactly what he means with disease subtypes.
Maybe different stages of the disease?
The problem with that will be the annotation. So because you don't know those different subtypes within a disease, and then you don't have the annotation for that. So we cannot predict in the way we are doing. But what we could do is, using experimental data, we can stratify the patients in different groups, and then we can try to see which profiles are similar to what diseases, and that might reveal differences in one disease. I don't know if that's what.
Yeah, I think that maybe is if you are working with diabetes type 1 and diabetes type 2.
Yeah, that's the hope, that you can definitely distinguish different diseases, but the hope is that
that's the idea.
We can identify also similarity between diseases that are comorbid.
Okay, so Kaula say, very nice talk. Did you try longitudinal multi-omics data integration to detect temporal relationships?
So as I was saying, you can add this time variable into the graph by using these time point links. And we have analyzed temporal data, but one modality only. So I haven't worked with different modalities and different time points. It was complex enough to use one modality.
Okay. Okay, second question from Kaula. Will our research go towards the multi-omics integration, or we will realize that most of the time is devoted to solving the data heterogeneity problems, and we will go back to single omic?
I don't think we will go to one single omic, and we shouldn't probably, because I really believe that different omics provide different information, and by taking all this information and interpreting all this information, we will be able to understand the complexity of some biological mechanisms. But it is true that in some cases, depending on the data that you have, it might be better to focus on one and then try to compare results, or focus on every single modality and then try to combine them, than taking everything together. But it has to be defined by its specific case.
Yeah. Great. So, any other questions from attendees? I think that it was very clear, your talk, and so let's wait a second.
Also, if anyone has questions, they can write me in the email.
If you want to talk about the open positions, what are the open positions that you have now?
Two postdoc positions, one here in Copenhagen and one in Oxford. And we are still defining the projects, but the one in Oxford will be in collaboration with some other departments at the University of Oxford, and that are working with transplant, kidney transplant and liver transplant data.
Okay, cool, Alberto. So thank you, Alberto. It was really nice for initiating our seminar webinar series. So just to remember that we will have 2 webinars per week on Tuesday and Thursday. Next Thursday we will have another webinar. So thank you, Alberto, and we hope to have you again in the future. So thanks. Bye. And thank you all.
Thank you very much.
Thank you.